Okay, so now we're going to continue with the statistical methods to cluster the fossil and extant taxa. But basically to explore your data, you already know your fossils, you know the ages, and you score all the morphological traits for the extant and your fossils. What do you do now? You go directly to infer the divergence times? No, let's do some exploration first. So that's what I was going to say that. So yeah, so for example, these are some seed fossils. As you can see, we were discussing about the different characters that we can score in them. And these are seed fossils. We have some internal structure to see the embryo of some of them, as well as for the extant taxa, how is the embryo and how are these testal cells and the walls of it. So now we are going to do some exploration. And something that we can do is, for example, take a phylogeny that was already published for our study group for with molecular data and then reconstruct the morphology of a particular character. So for example, we can use stochastic mapping for discrete traits, and we can see in that way how informative are the different traits. For example, here, this is of course a tomato family, a part of it. And you can see here the embryo shape and how there is phylogenetic signal mostly. Most of these, like some clades have this coil embryo, as well as some others have this straight embryo. So that could be informative in multiple ways. We can see if our characters are homoplacious, if they have multiple origins, and could give some noise to the analysis later. We can see also if, for example, we can trace this user of who is scoring the character, and in that way check if there is some problems with the scoring of particular people. We can also see this with continuous traits. So for example, this is seed length, and you can see that how the smallest seeds are in these clades, and then we have all of medium size. So we can do maximum likelihood reconstruction, we can calculate lambda, and we can do all this in and see how our characters are behaving. And then we have ordination techniques. So as you know, the ordination is this collective term to for multivariate techniques to reduce the data set, a multidimensional data set, in a way that projector in a low dimensional space, and in that way see how are the patterns of the data. So some basic ordination techniques is the principal component analysis, PCA. And as I said, these techniques are to reduce the dimensionality of the data, but also preserving as much as possible of the information contained in the original data. So the PCA, what it does, we plotted all this data, we have the variables. So use a rotation of the axis to maximize the variance in the data set. The PCA is a nation analysis. So there is a unique solution and uses Euclidean distance. We cannot choose the distance that we are using. And also the axes are orthogonal to each other, so they are independent. The distance between the points in this derived configuration, the two-dimensional, are proportional to the dissimilarities. And the axes are ranked by the eigenvalues, meaning that the first axis is the one that explains most of the variance of the data. The second axis is the second that explains the highest variance of the data as well. And potentially there are a large number of axes, but we don't need to specify the number of dimensions in advance. And the variable loadings of the original variables of the PCA, so might be understood and how much each of the variables contributed to building the PCA. But what we are going to do today during the practice is the non-metric multidimensional scaling. And what are the differences? So this analysis, the NMDS, relies in rank orders, meaning distances for the ordination. And as the same as the PCA, NMDS attempts to represent the pairwise dissimilarity between the objects in a low-dimensional space. So it's the same, trying to simplify the way that we see our data. But in the way that is different to the PCA, any dissimilarity coefficient or the distance measure can be used to build the matrix of dissimilarities. We don't use Euclidean distance. We can use any other according to the data that we have. So while the information about the magnitude of these distances is lost, these kind of methodologies, the rank-based methods, are more robust to data that they don't have an identifiable distribution. And because of we choose the dissimilarity coefficient, these type of methodologies accept a variety of different types of information. We can use continuous characters in PCA, but also discrete characters, for example. This methodology started in community ecology, but over the time was increasingly used to understand like fossil diversity and also taxonomic affinities. Also a difference with this methodology, the NMDS, is not an Asian analysis. And that means that there is no unique coordination result, which means that we need to run it several times to see if the result is stable. The axes of the ordination are not ordered according to the variance they explain, meaning that the first axis doesn't mean that explains most of the variance. The number of dimensions, we need to specify that once before the analysis. So a good practice is running this algorithm multiple times to ensure an ordination is stable, because we don't have a unique solution. Sure.
Are not ordered. How do you decide which ones you like if you wanted to plot it out? Like,
Okay, so the benefits about the non-metric multidimensional scaling is that it's a rank order approach that you can use any type of data, and uses a specialized distance matrix designed for the data, so it's more specific. And the drawbacks are that the dimensionality of the ordination space have to be specified a priori in advance, but we can do some tests to assess which number of dimensions is the best for our data. And also, another drawback is that it's an iterative process that may find a local, not global solution, optimum solution. But in that way, we have to run it multiple times. So it's a robust technique because tolerate missing pairwise distance. We can have missing data. Can be applied to a dissimilarity matrix with any metric. So can use quantitative, semi-quantitative, or mixed variables. And as I said, it's an iterative algorithm, and it has the following steps. So first, define a random placement of the data objects in this ordination space. After it does that, so specify the number of the reduced dimensions that we're going to use, and construct an initial configuration in the data in 2 dimensions. Then compare the distance between this initial two-dimensional configuration against the calculated distances, and then determine the stress of the data points with that. The stress of is the value that is the good fitness of the analysis. So reflects how well the ordination is summarizing the observed distances between samples. We want to reduce the stress as much as possible. And then according to that stress is correcting the position of the points in 2 dimensions to optimize the stress. So as I said, we have to establish the number of dimensions in advance. So what we can do is a plot of stress versus dimensionality to assess the proper choice of dimensions. We can do this according to the number of dimensions and how is the stress. And usually a stress that is less than 0.05, it's an excellent representation in the reduced dimension. Or if it's less than 0.1 is great, less than 0.2 is good, okay. And if it's between 0.2 and 0.3, it's not very good representation. So how are we going to do in R this? That is the practice that we are going to start in a bit. First, we're going to construct the dissimilarity matrix that we are going to use this package, this function daisy of the package vegan. And as I said, any dissimilarity coefficient or distance can be used. So in this package, we can include any of these 3. And we're going to check in a bit why any of these will be more useful, or according to our data, what will be more appropriate. Then we're going to run the non-metric multidimensional scaling, and you will see like in each run, the stress value for that run. And we're going to establish also the number of replicates that we want. And then we're going to feed the vectors, morphological characters, onto the ordination. So in that way, we'll see how are the characters informative and correlated to each of the axes here, which are informative, like are significative informative, as well as not informative at all sometimes. And the scores, we are going to give some similar information about that, about the weight of the characters in each of the axes. So this is mostly the data points outputs that are pulled to optimize the stress in multidimensional, the multi-dimensions in 2 dimensions. So we can also do this graphic of stress. And in this way, we can express it in ordination distance versus the dissimilarity observed. And this is called Sheffer plot. So what it does is with the stress, this goodness of fit measure, we can see that ideally all these points should fall into a single line, and increasing the ordination distance will be increase the observed distance as well. So we can do for each of the dimensions, having different stress values, and also doing the stress value according to the number of dimensions. That is here. Here we can have the stress versus the number of dimensions. And finally, we do the plotting, that there are already packages with the cluster, the functions within the cluster package to do the plot, but usually also ggplot, it's better for that, have more graphics, aesthetics. Okay. So one example is that we were seeing over the different talks is that the one of the seeds. So we can assess how our cluster the fossil seeds with extant seeds. And for that, I was also using some continuous additional characters that I'm not scoring for the 1500 taxa, but at least for 150. That is, for example, how are these cells in the middle? That are the testal cells, how is the area, perimeter, and roundness. So I did this analysis with 83 fossil records and also 280 taxa, extant taxa. For fossil records, I didn't include all the fossils that were assigned to that species, even holotype, paratypes, other specimens, everything. As you can see here, this is the analysis of the morphological characters, so it's 13 in total. And in red, you can see the fossils, and in blue, the extant taxa. And this was very surprising in the way that, for example, some fossils that were described as a single species were different species. And also the ones that cluster all together probably belong to one single species, and we can see in that way what would be the stratigraphic range image. Also, some of the ones that, for example, here, this is one physalis fossils that was described in Germany, but it clustered with one extinct fossil, nephrosemen, that was described here in America. So we found all these different differences between and clustering between fossil and extant taxa. Other example is, for example, this seed was described as Nicotiana from Russia, and this is Nicotiana, the extant taxa. So it doesn't cluster with it, it's something else. So we had multiple examples, and this, in that way, this analysis is not giving information only about the clustering between extant and fossil taxa, but also about your fossils and if they belong to the same taxa or not. So it's a good way to explore all the data that you are collecting. And with that, practice, coffee, and then practice. Questions first.